Okay, so welcome to today's R&D session with the Cybin team. We're here to talk about the progress we've made over the last 12 months to transform psychedelics into therapeutics. And at Cybin, we're on a mission to engineer transformative psychedelic therapeutics to improve mental health for patients. Working with a network of world-class partners, our internationally recognized scientists, we're harnessing already known attributes of classical psychedelics like psilocybin, and we're engineering them into commercially viable therapeutics that are accessible to all those that need them. Founded in 2019, we are a leading biopharmaceutical company because we're focused on the complete view of our ecosystem. We're engineering proprietary drug discovery platforms, innovative drug delivery systems, novel formulation approaches, so multiple approaches to the molecule, and treatment regimens for mental health, such as our EMBARK psychotherapy program. At a time when COVID-19 has exacerbated the mental health crisis to epidemic proportions, the Cybin team has been exceptionally productive. We have evolved in many ways in our journey to create ethical and equitable scientific platforms over the last year. And really, over the last 12 months, we've made tremendous progress in evolving our ecosystem. We've gained FDA approval for the first of its kind neuroimaging study with psychedelics. We've sponsored the University of Washington Psilocybin study for frontline COVID-affected healthcare providers. We've developed over 50 novel molecules with 15 patent filings across 3 patent families, and that patent estate just continues to grow. In just the past 12 months, we've completed 74 preclinical studies across our programs and established 50 professional partnerships with world-class scientists and advisors and clinical research organizations. We've created our EMBARK psychotherapy program to support both therapists and patients. And we've grown the company from 5 employees to 55 employees across 4 countries. We're operating today in Canada, the USA, the UK, and in Ireland. And we're the first and only company in this sector to qualify for a listing on the New York Stock Exchange. The team's done really a tremendous job. So we're excited today to share deeply meaningful scientific progress that we've made on the advancements to create promising approaches for patients in need of effective and safe prescription therapies in the mental health space. Cybin's comparative preclinical data clearly shows multiple potential advantages over classical psychedelic molecules like psilocybin. These data and the molecular possibilities may have wide-reaching implications for the treatment of mental health. And all of us here are aware of the enormous unmet need in this space. The World Health Organization indicates that as many as 700 million people around the world suffer from some form of depression or addiction or eating disorder. 300 million people globally suffer from depression alone. Alcohol use is the 3rd largest preventable cause of death annually, killing 3 million people a year. And anxiety disorders are really wide-ranging, with a prevalence as high as 12% of the population, so making them the most prevalent mental health disorder globally. And current treatments are lacking in efficacy and side effect profiles, so we can do better. But let's specifically talk about the challenges of oral psilocybin as a therapeutic. I think we're all excited about the academic studies that have shown efficacy with psilocybin, but it does have significant limitations. And Cybin is a biopharmaceutical company. Our goal is to transform these molecules into useful, practical therapeutics and get them through regulatory pathways. High variability in plasma levels means potential for unpredictable patient outcomes. Often patients don't see effects for the first 45 minutes of dosing, and maybe peak effects in an hour and a quarter or so. So patients are waiting for quite some time for the psilocybin to kick in. Long duration of effects with patients experiencing 8-hour clinic days. And some patients experience nausea. And all of this increases the time and resource burden on patients and providers and payers, limiting scalability and accessibility. So as is customary for any biopharmaceutical company, we recognize the challenges of the molecule, and we've worked in ways to address them by simultaneously looking at the formulation of psilocybin in 2 programs and then the molecule itself in a third. So we've had 3 parallel programs working on psilocybin for the past 12 months. So we've evolved our knowledge of oral psilocybin from a psychedelic to a therapeutic, and we've been developing, in the first 2 programs, CYB001 and CYB002, different oral psilocybin formulations. CYB001 is a sublingual film formulation designed to improve onset of action, meant to be faster. And we've done extensive work to optimize and improve membrane permeation. We've made a lot of progress in how we get psilocybin across the sublingual membrane. We've selected a lead prototype, and we've made clinical study supplies. But we now know that that film has to sit in the mouth for about 10 minutes, which may work, but we can do better. That's our thinking. CYB002 is a fast-dissolving oral psilocybin formulation, and additional research has resulted in a proprietary fast-release oral psilocybin formulation that releases psilocybin in just seconds. But the product has a long duration of action. So as you can see, we've tried different approaches and trying to tackle the various challenges with psilocybin. But what we're here to talk about today is CYB003, and we've developed a novel oral psilocybin analog called CYB003. And through the combinations of our learnings with CYB001 and CYB002, we've developed a molecule that appears to be efficacious. It's a deuterated psilocybin analog from our program, and I'm going to hand over to Dr. Palfreyman to take you through some of the data today, and then we'll summarize at the end. Thank you, Mike.
Thank you, Doug. So I'm going to show you data that supports addressing the problems that psilocybin has already been identified as having in a way to improve better outcomes for the patient. And Cybin's focus is improving this as a therapeutic, and obviously we want to use that as an oral drug. So the oral psilocybin analog has been designed to have less variability in plasma levels, to have a faster onset of action, to have a shorter duration of action, and to have actually better brain penetration, which is, of course, where it works. And we've done many different studies in a number of different animal species in the preclinical studies with models for psychedelic action, studying pharmacokinetic profiling, and then following the safety regulations that the FDA requires for advancing a program towards eventual clinical use. And we've done all of this to predict an efficacious and effective and safe human dose. So the next slide is going to show you some data which we've generated where we're measuring the concentrations of the active agent in the plasma, the blood plasma, against the time after an oral dose of either psilocybin, which is shown in the beige-colored line, or with CYB003, which is shown in the blue line. And the vertical bars on this are showing you the range of concentrations that you get following the dosing, and I'll explain that in a little bit in a minute. But what we're looking at here is the possibility that Cybin CYB003 would have much less variability than psilocybin, which, as we've explained, has been the problem with it. So if you take those, so if you like, the variability, and you show it in the figure here, you've got the large area in the beige color, which is psilocybin, and then below in blue is the signal that you get and the concentrations you get following CYB003. And you can see from the graph that the size of the variability is considerably smaller when you use the new compound CYB003. So in turning this into a actual measurement, we have at least 50% less variability with CYB003. And as we explained, that could translate to a safer dosing and potentially more predictable patient outcomes. Right. So that was the first part of the study we looked at. The second part we were looking at was whether or not we could get a faster time to onset. Again, psilocybin has a quite delayed time to onset, and so we looked at the, again, with these blood concentrations of the active agent, looking at the time to those. And so from this data, we were able to determine that the time to onset to clinically effective concentrations was at least twice as fast with CYB003 relative to oral psilocybin. So again, we've addressed the second concern that we have with psilocybin. Now the last point, which is the question about the very long duration of action of psilocybin, has also been addressed by looking at the time that we have concentrations that are therapeutically effective. And you can see from this graph that we do have a shorter duration of action. So for example, by between 2 and 3 hours, we've dropped below the level of therapeutic efficacy, and by 4 hours, we have almost no drug that's measurable. So in comparison, the psilocybin, even out at 4 hours, still has very high concentrations of active agent in the blood, and corresponds, of course, to the long duration of action of psilocybin. So if you just, again, do a calculation on the time we've saved here, we've actually reduced the duration of effects by at least half. So we've cut the duration in half, and that should result in much shorter clinical days, and of course that translates also into reduced costs. So one other very important advance we made with CYB003 was to establish that it also has a very much improved brain to plasma ratio, meaning more of the drug is getting into the brain, nearly twice as much compared to psilocybin. And this could lead to obviously lower doses because you need the drug in the brain, but also possibly less things like side effects like nausea. So if I sort of put this together on a sort of a summary chart, we know that psilocybin works as has great potential to work as a therapeutic. We've got very good preclinical data confirming that CYB003 will also have very similar, if not identical, therapeutic effects to psilocybin, with the advantages that it'll have lower variability, so you'll get safer dosing and more predictable outcomes. You'll have a faster onset of action, so less downtime waiting for the psychedelic action to come in, and obviously shorter duration of the study will have a great advantage in terms of reducing the overall clinical time as well as the costs overall. And again, as I said, the rapid and very effective brain distribution could allow for lower doses. So the potential is for the patient is to reduce the time and resource burden for both the patient and the providers, and obviously for the payers, and then improve the scalability and accessibility of treatment for these future patients. So I'm going to pass on now to Aaron, who will now tell us more about the next steps.
Thank you, Mike. Building on these very encouraging data that we have an obligation now to all patients that could benefit from this treatment to rapidly advance this molecule through the development phases. And we're positioned very well to do this. As Doug alluded to earlier, we've built an extraordinary internal scientific team with excellent support functions. But in addition, as he alluded, we've procured over 50 professional partnerships that again supplement and complement our internal acumen. So we're very excited about the abilities that we have to progress this molecule rapidly through the development phases. And you can see this already with the preclinical data that we've generated. We will have all of our preclinical studies, full package completed in the first quarter of next year. They will total 32. Our chemistry, manufacturing, and control development also is on the same path to conclude in the first quarter of next year, which includes the preparation of the early phase clinical trial materials. We will be filing an IND with the US FDA and clinical trial applications with the Medicines and Healthcare Regulatory Agency in the UK in quarter 2. And we will then progress upon regulatory approval our early phase studies in major depressive and alcohol use disorders, leveraging again, as was mentioned, our proprietary Embark psychotherapy program, subsequently then executing those trials in the US and UK respectively. So we're excited about the asset that we have. We have the capabilities to progress this molecule rapidly, and we look forward to continuing to share additional results with all of you. Back to you, Doug.
All right, thanks, Aaron. So yes, we've made some great progress, and obviously this data is really, really encouraging, but it's not just about the molecule. It's really about having the ecosystem and infrastructure around the organization and with our partners to continue to move these forward efficiently through the clinic. And I think you've seen over the past 12 months, this is exactly what we've been building. And Mike did a great job of summarizing the data, but at the end of the day, this is about patients. So why are we doing this? Why are we trying to optimize psilocybin as a molecule through our deuterated analog? We're doing it to improve the properties, improve its characteristics so that there's a benefit to patients. And the potential benefit for CYB003 for patients with MDD or AUD include 50% less variability in plasma levels, which means safer dosing and more predictable patient outcomes. We've seen even in academic studies that 2 patients given psilocybin might have a very different effect, a mild effect or a profound effect on the same dose. And if we can remove that variability, then it's a safer, more predictable treatment for depression and alcohol use disorder. CYB003 offers a 2 times faster onset of action, which means less downtime, less time the patient is waiting to get started with their treatment, and a faster onset of effects. And a 50% shorter duration also means shorter clinic days and reduce therapy and infrastructure costs around the molecule. And better brain penetration means therapeutic effects of CYB003 at lower doses, which means potentially a less risk of nausea and other side effects, which is always something we're aiming for. Less drug you can give for the same benefit, the better. So CYB003 presents an opportunity to combine our MDD program and our AUD program into a single program, protected by a family of 9 patents and growing, and resulting in total overall cost savings and efficiencies. By combining the 2 programs into one molecule, we're actually going to be saving, I think, somewhere in the range of $20 to $30 million over the course of development because you're not having to double work. And that means that the programs will be more cost effective and clearly the right path to take for our investors. And as you can see from, as Aaron demonstrated, we've got a rapid path to human studies in just the next couple of quarters to start dosing in man. So we're on a mission to make ethical and safe scientific progress to advance the care or treatment of mental health patients. And our goal has always been to be a leader in creating the best therapy for patients. And today, I think we've really made one step closer to making that a reality. So thanks to the team up here for presenting, and we're happy to take a few questions before we wrap up. Thank you. If you raise your hand, Leah's got a microphone.
Just a quick question. If you could explain a little bit further about the effects of the drug on its half-life, or usually deuteration would extend the half-life of a drug, so it's just a little confusing without a sort of explanation.
Right. Let me try and explain a little bit. There are lots of different things that deuteration does in addition to an effect on the actual half-life. So what we've seen in this particular case, very selective deuteration, we have an effect on the bioavailability, which is improved, the CNS penetration, which is improved, and also it has actually reduced the variability. So it impacts on those properties that we're looking to improve. And so deuteration has, depending where it is, does have multiple effects on the actual properties of the molecule.
And just to add to that, of course, we've started with an analog of psilocybin, not psilocybin itself, so already something that's short-acting. So, okay. Next question.
Hi, it's Pat Trucchio at H.C. Wainwright. Just for Doug, just a big picture question. You've built this incredible team in a short period of time, so I'm just curious, where are you in building out the Cybin team, and where do you see the space now as we stand? Is it, as you go to start enrolling in phase 1 trial, where's the enthusiasm level of getting study enrollment, patients, KOLs, et cetera, the entire ecosystem as you see it?
Yeah, you're right, Patrick, and thanks for the question. There is a lot more to drug development than sometimes people realize, and we spent a lot of time building strong partnerships, and those 50 or so partnerships cover chemical synthesis, preclinical study, work in multiple CROs in different geographies, and the early clinical infrastructure to start phase one studies, dosing studies. We have now supply chains available to us for our novel molecules in both Europe and the US, so we can work in parallel in 2 jurisdictions. That's really important when it comes to moving around a Schedule 1 drug. We need to do the work where we're making it, so we're able to make it in 2 locations. We have preclinical units established and set up with partners in both the US and the UK as well. So we'll be able to start phase 1 work in both locations as well. We've already started the process of reaching out for scientific advice from regulators. We expect to take those meetings early next year. And so we've got all the pieces in place. Aaron mentioned there's a 32 preclinical study package necessary. We've got about 10 remaining studies that are ongoing today. They're already contracted and the materials are available. So we're on track for the timelines that we just laid out. Thanks.
Thanks. Just a question. This is Sapir Manacheri from 8 Capital. I'm the healthcare analyst there, and I cover Cybin. Just noticed that the CYB program, you've now expanded the indication, the CYB003 program beyond AUD to also include MDD. Just wondering if that has an impact on your CYB1 program that has been focused on MDD, and just an update on where patient enrollment's at there, or if this is really all folded together.
Yeah, thanks, Seb, for the question. So I think externally we've been communicating CYB1 and CYB3 as separate programs. Internally, we've really been looking holistically at CYB1, 2, and 3 as to try to find the best approach to MDD and AUD and other potential indications that are coming, and doing that work in parallel. I think we got some great release results with CYB2. We've continued to optimize CYB1 and made a lot of progress, and we're ready to go with dosing. And then the CYB3 data, frankly, just blew us away. It checked all the boxes. CYB1 and 2 hit some of the boxes. CYB3 hit all the boxes. So we're still now a couple of quarters away from starting dosing studies, which is really not very far. But we think with a superior molecule, at the end of the day, will be better for patients and payers and providers. And ultimately, at the end of the day, this is about accessibility, which translates to market share. So I think we're going to end up capturing deeper use and more value over the long run. And as I said, combining the programs also creates an opportunity for cost savings. $20 to $30 million of saving from not having to run 2 sets of safety databases is very meaningful.
Understood. And does that translate also to the formulation side of it? Will you still be taking 3 through a oral film, or is that a cap on the formulation?
So the question was about formulation. CYB3, these are the data we showed as the raw material, right? So we've shown that the molecule itself has some inherently really strong properties. Great bioavailability, fast onset of action. So we still have an opportunity to optimize it further through formulation if we wish. So as we go through human studies and we see what the therapeutic outcome looks like, we could tweak that with making it faster, we could tweak it with making it longer. We have a lot of flexibility with formulation because the underlying molecule has great properties. So we haven't decided on a final formulation yet. We'll continue that work in parallel to human studies. We actually had made all of the materials ready to go with the CYB1 study, and the CYB data is here at the same time. So given the short time difference, only a couple of quarters to get to that dosing, we're prioritizing CYB3. Yeah.
How has the pandemic impacted the development cycle? Was it like beneficiary to be stuck in the lab, or do you think you'd be closer to human trials at this point if it weren't for it?
Well, this work on the underlying molecule, because it's novel, it just has to be done. Ultimately, you've got to get the preclinical studies, animal studies done for FDA and other regulators to let you start dosing in humans, and that's the right thing to do. Let's make sure it's safe before we do that. There has been a bit of a slowdown generally, nationally, globally, with preclinical studies. There's been a shortage of animal subjects, frankly. But as you can see, we've performed 74 clinical studies in the last year because the team has adapted to that, found great partners, and we continue to do that. So it hasn't really slowed us down. But the pandemic has affected all of us in some way or other. I think we've actually been benefited by being pretty virtual and being able to be pretty flexible through that process. Thank you.
Hello, Nicole Hodges with the Dales Report. Can you provide some more insight into the regulatory path?
Sure. So we expect to kick off phase 1 studies with dosing, as we mentioned, in the UK and the US. We'll then have to, once we've figured out the dosing, move into a proof of concept study. We're likely to prioritize MDD as the first indication. We think that has the greatest path and fastest path to successfully showing proof of concept. And then as we speak to regulators through that process, we'll determine what phase 3 will look like. And we fully expect, as this is a novel molecule, that we would qualify for NCE exclusivity. In the US, we would certainly want to take advantage of any breakthrough therapy designation or fast track designation that's available, given the potential impact of this molecule on these indications.
Thank you. And one more question. What kind of IP protection do you think you're going to be able to achieve?
Well, the IP is continuing to expand. This particular family of patents, our tryptamine family, we currently have 9 filings. And you'll start to see some of those become public in the near future. We still have work to do as we optimize the molecule and as we complete preclinical and clinical studies. That'll generate more data. So I certainly expect that patent estate to continue. Thanks.
There are many companies in this space. What do you believe are the competitive challenges that Cybin faces?
That's right. There are a lot of companies working on psilocybin. I think ultimately not all of those psilocybin molecules products will come to market. But this is a new area of treatment. There's still a lot of education and training to do, the general public, for therapists. These are very large populations suffering from MDD and AUD. So I think there's an opportunity for multiple approaches to these conditions. I don't really see at this stage that we're dealing with a range of competitors, but options for patients ultimately when we come to market. Leah Elmer down here has a question.
Thank you. Thank you, Doug. So how would the 003 be different theoretically from deuterated psilocin, if you were to imagine deuterated psilocin's profile?
We would have to do that work to determine.
Okay. Yeah. So it's not deuterated psilocin.
It's a deuterated analog of psilocybin. I appreciate the question, Elmer, but we're in a delicate IP situation right now, and also competitively, we've seen in the past as we've made certain progress, some of our competitors have followed, and so I think at this point, while the IP landscape is still emerging, we'll want to keep that proprietary for now. Ultimately, there'll be a final formulation of this molecule that we will decide upon. So at that time, we'll be able to share exactly what it is. Okay.
Hi. What is left to be done in the preclinical trials?
Do you want to say that one?
Yes. So we're at the final stages of doing what's called GLP studies, which are the FDA-regulated studies, and those consist of looking at the safety and toxicology. We have all that information sort of ready to go, but now we do it with this official FDA-mandated set of trials, and that's all there is really to do. So the drug is available, everything else has been done. So
I would add, though, that in the early toxicology studies that we've done, we've seen no differences to psilocybin.
No problems at all. Yes, exactly. So we don't have any issues. We just have to complete the work.
Two questions. Are you just targeting depression, anxiety, and alcohol addiction? Those are the 3 that you mentioned earlier. Are you looking at other types of disorders?
I love the question. Are you only tackling?
All right, let me rephrase that. Okay, first of all, you're picking on someone who has not had her coffee this morning. So that's my addiction.
I can answer your question. So
Okay.
we're starting with major depressive disorder. There may be benefits in treatment-resistant depression later. We'll save that for later. We're starting with alcohol use disorder, but there may be potential in tobacco use or substance opioid use disorder. We're starting with generalized anxiety disorder and social anxiety disorder, but there are many other anxiety disorders. So we're starting with a core proof of concepts in these first indications, and we may see an expansion either through work that we do or through that is done in academia.
Okay, and then the second, I should have used the word primarily. I really apologize. And then, would this medication be used in lieu of or in addition to the existing antidepressants, whatever, on the market now?
I can give you my personal opinion on that. I'll tell you that in our clinical studies, I expect that patients will have to come off of their existing, say, depressive treatment. You have to kind of separate our active drug from something else that they're taking. So in clinical studies, that's likely to happen. But it may be that as we learn more about these treatments, that there might be an opportunity to combine, and patients may take SSRIs and have intermittent therapy with psychedelics. But I don't think we're quite there yet with the data to really be able to answer that question.
Hi there, Amanda with Forbes. I'm wondering also about the indications that you're looking to treat. So why did you select major depressive disorder, alcohol use disorder? I'm just curious about that.
Yeah, I'm sure it's a very lengthy process, actually, is to go through that sort of exercise of determining which indications. Clearly with psilocybin, there's a lot of background data, academic data in depression. So that's a good starting point. We can define quite clear clinical endpoints that would be acceptable by regulators in both Europe and the US. We can define for MDD a preclinical pathway and a clinical pathway. So those are really the criteria. And then obviously the market potential and those other things at the end. Those are the criteria. We feel the same with AUD. We felt the same with social anxiety disorder, general anxiety disorder. But it is a lengthy process. It's not a simple thing.
Hi, this is Frank Breeswell with Oppenheimer. Just a couple questions. You touched on quickly the EMBARK program, and I was wondering, so you're going to use that program going into clinical trials, and how do you make sure that you can deal with the variability with using a psychotherapy training in the trial?
Hey, Frank, thanks for the question. Didn't recognize you with the mask on over there. So we developed EMBARK specifically to deal with variability. This is an unusual situation. It's new ground to combine a drug with psychotherapy and take it through the regulatory pathway. So we wanted to make sure that the psychotherapy was as consistent as possible. So we created EMBARK from currently existing best practices in order to apply consistent training for therapists. And so that those therapists could then apply consistent therapy in the clinical studies. And I'll say that we're making EMBARK open source. We're making it available to therapists that want to be trained. We want to make sure we don't limit access to that. And we hope that we can use the program not only to support our clinical studies, but to educate and train therapists over time as well.
Okay, and then just lastly, on the duration of effect, what are your thoughts on the fact that maybe to get a clinically meaningful effect, you need to have a long duration of effect of the drug for a classic psilocybin product?
Yeah, I'm not sure that that's been proven. I think there's good evidence that ketamine has benefits in depression with a much shorter duration. There's evidence that ketamine with associated psychotherapy is better still. So clearly there are molecules out there that indicate that a few hours may be enough. And we'll see when we get into clinical studies. It's a bit early to make judgments. We can see in the preclinical models a psychedelic effect, and our human studies will have to demonstrate that psychedelic effect turns into a therapeutic benefit. Maybe one more question? Leah.
Hi, James Halifax from the Psychedelic Spotlight. I just had a quick question on the production process of CYB003. I'm curious if it is a resource-intensive process, or once you have the expertise and know the know-how how to create it, whether it's fairly simple to synthesize.
Do you want to say that?
Yes. So it's very much a classical chemical synthesis with proper controls and everything. Number of steps that are in the synthesis are very standard. So there's nothing here that is in any way slowing down development of the product and scaling it appropriately.
And I'll just add to that, as I mentioned earlier, that we've contracted with suppliers in both Europe and the US to produce our proprietary molecule. So we've made sure that we'll have supply in both jurisdictions for clinical studies, and then we have choices, of course, when it comes to commercial supply. So I think we're well set up there. And with that, we'll wrap up this morning. Thank you very much for the questions and for your interest in Cybin. Thanks very much.
Thank you.